Good afternoon, everyone. This is Inan from Microbiosystems. Thank you very much for having me here. It's a great honor for me to have this opportunity to share with you the critical roles of antigens and antibodies in SARS-CoV-2 serological test development. I would like to talk about what Microbiosystems has done to help fight against COVID-19. Today, I will start my presentation with the introduction of the outbreak of COVID-19. COVID-19 pandemic is caused by the emerging coronavirus SARS-CoV-2, and it presents a serious global public health emergency. The first cases of COVID-19 were reported from Wuhan, China, in December 2019. According to the World Health Organization, the pandemic is affecting 213 countries and territories around the world and 2 international conveyances. As of July the 6th, over 11 million confirmed cases of COVID-19 have been reported, and over 0.5 million deaths have been caused by this pandemic. In the COVID-19 map offered by Johns Hopkins Coronavirus Resources Center, the red dots represent the confirmed cases of COVID-19, and the larger the red dots, the more confirmed cases in the specific region or country. And you can see that COVID-19 has spread to most parts of the globe. In addition to the health damage caused by the pandemic, COVID-19 has huge impact on our daily life. Now we have got used to wearing protective masks and keeping social distance in the public place. COVID-19 is caused by SARS-CoV-2, which belongs to coronavirus. Now let's take a look at the background of this virus species. Coronavirus was first identified by Dr. Almeida in 1964 in St. Thomas Hospital in London. They are a group of envelope viruses that have a halo or crown-like appearance when viewed under an electron microscope. The genome of coronaviruses are composed of positive sense single-stranded RNA, and their genome size ranges between 27 to 34 kilobases, which is relatively large for RNA viruses. Coronavirus infect mammals and birds and cause various symptoms, such as respiratory tract disease and diarrhea. This virus species can be characterized into 4 genogroups, including alpha, beta, gamma, and delta. And within the beta coronavirus genogroup, there are 4 lineages, including A, B, C, and D. 7 coronaviruses have been reported to be able to infect humans. Among them, HCOV229E, HCOV-NL63 belong to the alpha genogroup, and HCOV-HKU1, HCOV-OC43 belong to A lineage of beta genogroup. These 4 coronaviruses can only cause common cold. SARS-CoV and SARS-CoV-2 belong to B lineages, and MERS-CoV belong to C lineage of beta genogroup. These 3 coronaviruses have been shown to be lethal. SARS-CoV-2 spike protein is a key transmembrane glycoprotein that forms homotrimers protruding from the viral surface and mediate whole cell attachment and cell entry. It was reported that SARS-CoV-2S and SARS-CoV-S share the same functional whole cell receptor, which is angiotensin converting enzyme 2, also ACE2. Spike protein contains 1,273 amino acids and can be cleaved into S1 and S2 subunits. S1 subunit is responsible for binding to the whole cell receptor. It consists of N-terminal domain or NTD and the receptor binding domain or RBD. S2 subunit mediates fusion of the viral and cellular membranes, which contains fusion peptide FP, peptide repeat 1 HR1, or central helix CH. Currently, spike protein extracellular domain, including both S1 and S2 subunits, S1 subunits alone and RBD alone, have been widely used in the serological test development. We'll talk about the application of these antigens into details later. At present, the COVID-19 pandemic within the many countries have not yet reached its peak, and the number of cases and deaths are predicted to rise in the coming weeks and months. Early and accurate diagnosis of COVID-19 is essential, not only to ensure appropriate patient care, but also to facilitate identification of SARS-CoV-2 infected people to control disease spread. The etiological diagnosis of SARS-CoV-2 infection is regarded as gold standard for diagnosis and nucleic acid testing with real-time PCR has been widely employed. However, high numbers of false negatives have been found in some laboratories, leading to a positive detection rate of RT-PCR, around 50% of suspected clinical and epidemiological COVID-19 cases. In addition, nucleic acid testing is operator-restricted, time-consuming, easy to pollution, and expensive. Serological tests can be used to identify whether people have been exposed to a particular pathogen by looking for the presence of antibodies that are produced in response to infections. IgM is first elicited by the immune system in early stage of infection, and then IgG is generated in large quantity after 7 days of symptom onset, which can persist in the blood for several weeks or months. Serological tests offer some advantages over real-time PCR. Firstly, human antibodies are much more stable than viral RNA, so serological specimens are less sensitive to spoilage during collection, transport, storage, and testing than real-time PCR. Secondly, because antibodies are typically uniformly distributed in the blood, serological specimens have much less variations than viral RNA specimens, and it can be easily collected. Thirdly, unlike real-time PCR, serological tests can detect past infection because of the long-term existence of antibodies in blood. Therefore, serological test is an efficient supplement of nucleic acid detection, and the demand for serological tests of SARS-CoV-2 is increasing rapidly in order to improve the accuracy of clinical diagnosis and better quantify the number of cases of COVID-19, including those that may be asymptomatic or have recovered. Serological test kits consist of 3 types, including colloidal gold immunoassay, chemiluminescence immunoassay, and ELISA. Now let's take a look at the colloidal gold immunoassay. Colloidal gold immunoassay is a kind of lateral flow immunoassay. The test strip is made up of 4 parts, including sample pad, conjugate pad, nitrocellular membrane, and absorbent pad. Before testing, the gold conjugated antigen is placed on the conjugate pad. Anti-IgG IgM secondary antibody and anti-antigen antibody are put on nitrocellular membrane as test line and control line, respectively. When a drop of serum that contains IgG and IgM is put on the sample pad, the antibodies would move into the direction of absorbent pad. If the serum includes the IgG or IgM against the pathogen, these antibodies would bind to the antigens on the conjugated pad and then interact with anti-IgG IgM secondary antibodies at the test line. Hence, at the test line where gold labeled proteins is accumulated, red or pink spots can be seen by the naked eye. The colloidal gold immunoassay strip shown here indicates that the serum contains the antibodies against the SARS-CoV-2, and the patients have been infected by the virus. The colloidal gold immunoassay is easy to use, and it only takes 15 minutes to see the results. The list here shows the commercial colloidal gold immunoassay test kits that have received emergency use authorization from FDA. For the chemiluminescence immunoassay, it belongs to labeled antibody technology, and it labels antibodies or antigens with chemiluminescent agents or catalytic luminescent enzymes. When labeled antibodies or antigens are bound with corresponding antigens or antibodies, the luminescent substrates are activated by luminescent agents or catalytic enzymes, and then the released visible light or luminescence of fluorescent substances are detected by luminescent spectrometer. For ELISA, it employs similar principle as chemiluminescence immunoassay, except that it uses enzyme-labeled antibody or antigen. The main advantage of chemiluminescence immunoassay and ELISA is the higher sensitivity compared with colloidal gold immunoassay, but they usually need around 1 hour to get the result and require instrument or labs to perform the test. After talking about the application of SARS-CoV-2 antigen in serological test development, I'll be introducing what Microbiosystems has done to help fight against COVID-19. First, I would like to offer you a brief introduction of Microbiosystems. Microbiosystems is founded in 2010, and we are a leading manufacturer of recombinant proteins and other critical reagents to support the development of target therapeutics. Microbiosystems is dedicated to accelerating the process of target therapeutic drug development, from the starting point of target identification to endpoint of clinical trial. Microbiosystems has served over 3,500 customers from around 60 countries and reached out to 20,000 end users from pharmaceutical companies and academic institutes. Acro also established long-term collaboration with big firms such as Pfizer, Novartis, Johnson & Johnson, and well-known academic institutes like NIH. To provide better service to end users, Microbiosystems has set up 2 working places at Beijing and Shanghai in China, and 3 offices at Boston, San Jose, and Newark in US. With the outbreak of COVID-19 in China, Microbiosystems responded quickly and finished the design of SARS-CoV-2 antigen products during Chinese Spring Festival, and immediately started to produce the antigen after getting back to work. Up until now, we have launched over 40 SARS-CoV-2 antigen products, and another 20 products are under development. The majority of these antigen products are spike ectodomain, S1 subunit, and RBD proteins. Acro has worked hard on developing products that can be used in serological test development. We have made following efforts to provide high-quality products to our customers and help fight against COVID-19, including verifying the trimeric state of our spike ECD protein product, the production of biotin-related spike protein, optimizing the expression construct for spike protein RBD, developing anti-SARS-CoV-2 antibodies, and producing mutant spike protein RBD. First, let's see how Acro has produced SARS-CoV-2 spike protein in trimeric state. The trimeric state of spike protein is required to develop a serological test for COVID-19. NIH has published a paper describing the development of an ELISA kit for sero surveys of the SARS-CoV-2 pandemic. The trimeric structures of SARS-CoV-2 spike protein was first verified by analytical size exclusion chromatography and transmission electron microscopy, and then deployed in ELISA kit. To produce trimeric spike protein, we introduced a GS-AS substitution at the S1 or S2 fusion cleavage site to maintain the integrity of spike protein. In addition, a fold-on trimerization domain was introduced just in the upstream of the predicted transmembrane region to facilitate the trimer formation of spike protein. Microbiosystems uses its proprietary HAC293 expression platform to produce recombinant spike trimer protein, and the purity of the protein is greater than 90% in SDS page, and the protein migrates as about 200 kilodaltons under reducing conditions due to glycosylation. Since the purity on SDS page cannot indicate the conformational homogeneity of the protein, we performed SAC mouse to show that the trimeric S protein is with more than 80% purity, with molecular weight around 600 to 660 kilodaltons. To further characterize the bioactivity of the spike protein, we use surface plasmon resonance to measure the binding ability between spike trimer and its host receptor ACE2. The constant dissociation shows around 35 nanomolars, which is consistent with binding affinity reported in paper. In addition, we are the first company around the world to provide the negative stain electron micrograph of our spike trimeric protein. The result shows that the well-folded trimeric particles of SARS-CoV-2 protein are similar in size and appearance to previously characterized SARS-CoV-2 trimer. We also performed SAC mouse to compare our SARS-CoV-2 spike protein product with the protein offered by other vendors. As you can see from this slide, in contrast to Acro's trimeric spike protein product with a conformational purity of 97%, the spike protein offered by another vendor are in high polymer form with molecular weight around 4,500 kilodaltons. In addition to unconjugated antigen product used in serological test kits, the biotinylated SARS-CoV-2 recombinant antigen is also employed in commercial chemiluminescent immunoassay products. On May the 29th, Siemens Healthineers Diagnostics receives FDA emergency use authorization for its SARS-CoV-2 total antibody test. The total antibody test has demonstrated 100% sensitivity and 99.8% specificity in identifying SARS-CoV-2 antibodies. The antigen used in the antibody test is biotinylated SARS-CoV-2 recombinant antigen, which bind the streptavidin-coated microparticles and capture anti-SARS-CoV-2 antibodies in the specimen. Microbiosystems has developed an exclusive collection of pre-labeled and experimentally validated biotinylated SARS-CoV-2 spike proteins. These products are produced using the AVI tag technology, which introduces a unique 15 amino acid peptide AVI tag into the recombinant protein during expression vector construction. The single lysine residue in the AVI tag is enzymatically biotinylated by the collobiotin ligase BRA. In this way, the biotinylation only happens on the single point lysine residue of AVI tag. The protein orientation is uniform when immobilized on an avidin-coated surface. One of our customer drugs and diagnostics for tropical disease has commented after using our biotinylated spike proteins that AVI tag biotinylation technology from Microbiosystems as a great fit for our LFA platform. The next part is about the optimization of expression construct for spike protein RBD. Because recently a paper published by NIH has shown that different RBD constructs have distinct signal when used in developing serological ELISA assay. The two RBD constructs from Kramer lab of Mount Sinai and Reagan Institute were compared, and the construct from Reagan Institute displayed stronger signal with the recombinant RBD antibody. ACRO also got feedback from our customers that the appropriate RBD construct can determine the sensitivity and the specificity of ELISA kit. We are currently working on optimizing the RBD construct to increase the sensitivity while maintaining the specificity of the kit product. And these products will be launched at the end of this month. ACRO noticed that several RBD mutants have been reported in some strains of SARS-CoV-2, and we generated the mutant proteins products to characterize the difference between wild type and mutant RBDs. ELISA and SPR results have shown that the RBD mutants shown in this table can bind ACE2 with 2 to 6-fold higher affinity compared with wild type RBD. These mutant RBDs can be used to test the neutralizing ability of anti-SARS-CoV-2 antibodies to exclude the possibility that the mutation in RBD makes the protein unrecognizable for the antibody. We tested binding ability between our anti-SARS-CoV-2 antibodies and the RBD mutants. These antibodies also similar binding affinity with RBD mutants and wild-type RBD in SPR assay. In addition, the antibodies can effectively inhibit the interaction between RBD and ACE2. These studies indicate that the RBD mutants developed by ACRO are useful research reagents to verify the broad spectrum neutralizing antibodies against different strains of SARS-CoV-2. In addition to offer SARS-CoV-2 antigen products, ACRO collaborated with its partner to develop anti-SARS-CoV-2 antibodies as well. ACRO Biosystems has developed a variety of mouse and chimeric anti-SARS-CoV-2 monoclonal antibodies and mapped the epitope of these antibodies. As you can see here, there are 6 regions that is characterized by our scientists. Anti-SARS-CoV-2 S1 and N protein antibody pair has been launched for serological assay development. The sensitivity of detecting nucleic capsid protein by anti-SARS-CoV-2 nucleic capsid antibody pair in our ELISA assay is 12.5 pg per milliliter. Microbiosystems has developed SARS-CoV-2 antigen and antibody product specifically designed and optimized for serological test kits. Spike-derived proteins are produced from HEC293 cells, while nucleocapsid proteins are either yielded from HEC293 cells or E. coli cells. The antigens are expressed with a variety of tags, including his, human FC, mouse FC, and his AVI tag, which could meet the needs of colloidal immunoassay, chemiluminescence immunoassay, and ELISA development. Here is the list of our products specifically designed for serological test kits, and you can contact us if you want to know more about these products. Now, this will be the last slide of this presentation. Thanks everyone for attending today's webinar. We're doing a raffle for everyone. You will have the chance to win our adorable llama. They are very popular among our customers. If you have not already entered this raffle, you can go to our website, www.microbiosystems.com, and enter it. We will contact you within 3 business days if you win the llama. Okay, that's all for my presentation. And now I would like to answer the questions from the audience.
Yeah, thank you, Alan, for a great presentation. I'd like to open up the floor for questions. So please send them to the chat feature or raise your hand via the GoToWebinar platform. Thank you. Sure. So we have one question here. How do you ensure the serologic ELISA kit is specific?
I'm sorry, could you repeat the question?
Oh, yeah, sorry about that. How do you ensure your serologic ELISA kit is specific?
Because we have developed a variety of pairs of antibodies, and we have tested the species specificity of these antibodies. I have not shown the data here. We have tested if the antibody could react with like antigens from other coronaviruses like SARS, MERS, and like HCOV2. So the antibodies are species specific to SARS-CoV-2, and so the antibody pair is also species specific. So we are pretty sure that the antibody pair or the antibody alone is very specific for SARS-CoV-2 antigen.
Okay, brilliant, thank you. And the next question is, are you also providing spike protein variants of the other viruses as controls for the assay? Would this not be important? So question mark, yeah.
Yes, sure. We have used the negative control to ensure that the assay is convincing. And so the question from the audience is that if we have enough control in our assay.
Pardon, sorry?
So the question from the audience is that have we put very good control in our assay?
So yeah, I think the question is essentially, are you providing spike protein variants of the other viruses as controls for the assay? I mean, that's yeah.
Yeah. There is a popular strain in the Europe and US now. It has these 614G mutation in the spike protein. Now, we also use this protein in our assay, and the mutant spike protein shows the similar binding affinity to our antibodies and ACE2. So it doesn't show any very different prospect of this between the wild type protein and the mutant protein.
Okay, brilliant.
So thank you.
Sorry. Yeah. The next question: does every kit come with a positive control?
Oh yeah, because we have also developed, neutralizing antibodies with our partners. We isolated these antibodies from SARS-CoV-2 infected patients, and these neutralizing antibodies have shown potent, potent activity against pseudovirus and authentic virus of SARS-CoV-2. So we have used this neutralizing antibody as the positive control, and our antibody pair and also the serological test is very sensitive with our neutralizing antibody.
Okay, brilliant. And another question: can you comment on antibodies with broad spectrums to detect human coronaviruses?
I'm sorry. Could I comment on what?
Can you comment on antibodies with broad spectrums to detect human coronaviruses?
Okay. Yeah. We have collaborated with our partners to screen a lot of antibodies, and we have known that the antibodies isolated from SARS-CoV-2 infected patients are not, cannot bind to SARS-CoV antibodies. So these antibodies are species specific. But I have also, read papers that reported an antibody that isolated from SARS-CoV antibodies can react with SARS-CoV-2 antigen, but it's not very, it's not very many antibodies until now. So I think it's still a challenge for us to develop broad spectrum antibodies against coronaviruses. But we're still on the road. Yeah.
Okay, brilliant. And another question: do you have different pairs of antibodies for different strains of COVID-19?
Not really. We only, have antibodies from SARS-CoV-2 infected patients from China, and we also screen antibodies from immunized mouse and rats. But we just use, the same spike protein as the antigen to immunize the animals. So we don't have, pretty much different. We don't have many antibodies for different strains of, the virus. But we have tested, the virus, the antibodies reactivity between wild type spike protein and D614G mutant. So that doesn't show, distinct bioactivity.